Good morning! It's August 8th, and this is your daily brief in science. Here's everything you need to know. Researchers in Australia are taking a unique approach to ocean exploration by utilizing sea lions equipped with lightweight cameras to map previously unmapped ocean floor habitats. This project, led by PhD student Nathan Angelakis, involved 8 adult female sea lions from Olive Island and Seal Bay, outfitted with cameras and tracking devices. Throughout the study, these sea lions captured an impressive 89 hours of footage, documenting 6 distinct seafloor habitats, which include macroalgae reef, macroalgae meadow, bare sand, sponge/sand, invertebrate reefs, and invertebrate boulders. This research enhances the understanding of biodiversity in these regions by revealing previously uncharted areas of the ocean floor off the southern coast of Australia. Understanding these habitats is crucial for their protection against threats such as human activities and climate change. The researchers also validated a machine learning model that predicts seafloor habitats with over 98% accuracy, using the footage to assess biodiversity effectively. The findings also highlight the sea lions' strong knowledge of their environment, which will assist in conservation efforts for this endemic species. Professor Simon Goldsworthy, who has studied the species for 25 years, described the footage as slow TV, providing unprecedented insights into the sea lion's underwater activities. Notably, the footage includes the first observation of a mother Australian sea lion teaching her pup how to hunt, showcasing the species' social learning behaviors. These findings, published in the journal Frontiers in Marine Science, contribute significantly to the understanding of endangered marine species. Current methods for mapping seabeds, such as remotely operated underwater vehicles, are often costly and hindered by weather conditions, making this innovative approach particularly valuable. Researchers at the University of Stirling have unveiled a concerning prediction regarding tick populations in Scotland, forecasting that they could nearly double by the year 2080 if global temperatures rise by 4 degrees Celsius. This study, led by Professor Rachel Norman, highlights the potential health risks associated with rising tick populations, particularly the spread of diseases such as Lyme disease. The model developed by the researchers indicates a significant 26% increase in tick density with just a 1 degree Celsius rise in temperatures by the year 2080. While world leaders have set ambitious goals to limit temperature increases to 1.5 degrees Celsius, the study suggests that this may not be enough to curb the expansion of tick habitats. Areas that were previously considered tick-free, such as montane habitats, are projected to become suitable for ticks by the year 2080. The study predicts that woodland habitats will experience the highest absolute increases in tick density, while mountain slopes will see the largest proportional increases. The anticipated rise in tick populations raises concerns over a surge in Lyme disease cases, a bacterial infection that affects about 1 in 10 people globally. Lyme disease is often misdiagnosed and can lead to severe long-term health issues even with treatment. The methodology used in this research is dynamic and adaptable, allowing for its application to other regions and vector species. This enhances our understanding of how environmental changes impact populations. The findings were published in the journal Interface, under the title GIS-ODE, Linking Dynamic Population Models with GIS to Predict Pathogen Vector Abundance Across a Country Under Climate Change Scenarios. Given the increasing concerns in Scotland regarding tick abundance and Lyme disease, this ecological modeling is both relevant and timely. Recent studies reveal alarming trends in agriculture as environmental extremes like drought and flooding become more frequent, leading to significant crop losses. Research has shown that both drought and waterlogging adversely affect physiological parameters, particularly leaf gas exchange and stem water potential, with waterlogging demonstrating more severe impacts. Drought conditions have been linked to notable reductions in morphological growth, resulting in decreased plant height, leaf area, stem diameter, and root weight across various genotypes. In terms of photosynthesis, drought stress caused significant declines in net photosynthetic rates and stomatal conductance for all tested hybrids, although the hybrid EQ20 showed relatively better performance. Additionally, the quantum yield efficiency of photosystem 2, written as FV over FM, decreased by 43% under drought stress, with EQ20 again exhibiting the least damage compared to other hybrids. RNA-seq analysis highlighted that chorizo had 6,092 differentially expressed genes in response to drought, while bitters had only 320, indicating varying responses to drought stress. This research underscores the potential of utilizing gene expression markers for early detection of stress and differentiating between drought and waterlogging in kiwi fruit. Furthermore, the study explores the EPF/EPFL gene family in maize, which is crucial for regulating stomatal behavior, to better understand its role in drought tolerance. It also emphasizes the importance of comprehending the molecular mechanisms of drought tolerance in foxtail millet, which could influence future crop breeding strategies. Drought stress has been shown to impact several metabolic pathways, particularly those involved in plant hormone signal transduction, protein processing, and mitogen-activated protein kinase signaling. Transgenic plants that overexpress CYP67 demonstrated lower malondialdehyde levels and higher activities of antioxidant enzymes under drought conditions. These findings provide valuable insights that could help inform breeding strategies aimed at enhancing drought resistance in maize, contributing to agricultural sustainability in the face of climate challenges. A new study sheds light on an unexpected consequence of airlines flying at higher altitudes. While this practice has led to a reduction in carbon emissions, it has also resulted in an increase in the formation of long-lasting contrails, which may contribute to climate change. The study emphasizes that modern, cleaner engines produce fewer soot particles, allowing contrails to dissipate more quickly. However, the warming effect of these contrails could potentially exceed the impact of carbon emissions from jet fuel. Researchers analyzed flight data and satellite observations from over 64,000 flights to understand the persistence and warming effects of contrails, revealing significant challenges in studying this phenomenon. The research primarily focused on flights over the Western Atlantic, indicating that these findings might not be applicable to areas like Greenland and Iceland, where conditions for contrail formation differ. Professor Richard Allen noted that the increasing number of flights presents a greater challenge than the warming effects of contrails themselves. Dr. Edward Greispert, the lead author of the study, pointed out that the climate impact of private jets is often underestimated due to their disproportionately high impact per passenger. In a proactive approach, American Airlines is testing AI and satellite imagery to adjust flight altitudes, which has shown promise in reducing contrails during their test flights. Experts suggest that while adjusting altitudes might increase fuel consumption during descents, the environmental benefits of reducing contrails could outweigh these costs. Additionally, the study found that private jets produce contrails as frequently as larger commercial aircraft, raising concerns about their environmental impact. Overall, the findings suggest that the aviation industry's efforts to reduce carbon emissions are being undermined by the warming effects of contrails, highlighting the need for better regulation and alternative fuels. Recent research from the University of Copenhagen has shed light on the molecular interactions of cocaine with the dopamine transporter, or DAT. This study is particularly significant as it reveals how cocaine inhibits DAT, a crucial component in regulating dopamine levels in the brain. One key finding indicates that the compound MRS7292 binds deeply within the HDAT structure. This unique binding creates a pharmacological profile distinct from other transporters, such as HSERT, which may lead to novel therapeutic avenues. Using advanced cryo-electron microscopy techniques, researchers have mapped the binding mechanisms of various molecules to DAT. This detailed analysis enhances our understanding of the transporter's structure and function, which is vital for dopamine transport, leveraging sodium and chloride gradients for substrate uptake. Dopamine, often called the feel-good hormone, is regulated by DAT and is essential for mood, reward, and motivation. Cocaine disrupts this regulation, resulting in a skewed perception of pleasure, making it difficult for users to distinguish between pleasurable and non-pleasurable experiences. Professor Klaus Lowland has pointed out the potential for these findings to inform the development of medical treatments for cocaine abuse, which could significantly benefit those struggling with addiction. The overarching research goal is to create targeted pharmaceuticals for conditions linked to DAT dysfunction, including various forms of addiction. Harold H. Sitt from the Medical University of Vienna has emphasized the importance of these findings for future therapeutic applications. The implications of this research extend to understanding dopamine imbalances that contribute to several brain disorders, such as Parkinson's disease and psychological issues. Published in the journal Nature, this study underscores the significance of comprehending cocaine's effects on the brain and the potential pathways for treatment. For those seeking more information, additional details can be accessed through the linked paper. This has been your daily brief in science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.